its structure. One is its position within the body, second is what it's connected to, and thirdly is can you find any intermediate stages. Now Owen didn't change any of these criteria. What he did was formalize them with a couple of definitions that made it very easy for people to see these similarities. And so you have position and connections, the humerus of the human, the humerus of the seal, the radius of the ulna, the radius of the ulna, the digits. And so you could talk about homology of the limbs as forearms. You could talk about homology of the radius of the ulna as two elements. So you can have homology at different levels within these structures. So if two characters are not homologous, then what are they? Now Richard Owen said that, now remember Richard Owen was publishing this in the 1840s. He never accepted Darwinian evolution, although at various stages he claimed that he'd actually thought about it and written about it himself. And so these are pre-Darwinian concepts of Owen's. And so what Owen did was to contrast homology with analogy. Analogy is when you have similar functions but very dissimilar structure. So the wings of a bat and the wings of an insect serve similar functions, but they clearly are structurally very, very different. One based on skeletal elements of cartilage and bone, one based on a very different system. So this was not an evolutionary concept, Owen's concept of homology and analogy. Ray Lancaster, who became the second director of the British Museum of Natural History, because he was fired for being cantankerous and difficult and all sorts of other things, he introduced an evolutionary definition for homology in 1870. And he used two terms, and this is, there's a little bit of technical stuff here, but not much. He used the term homogeny, that's what we currently call homology, and homoplasy. And so for him, homogeny when there were a very close evolutionary history, and homoplasy when there was a more distant evolutionary history. So both these concepts had to do with degree of relatedness. Very similar, sharing a common ancestor, more different. Obviously we use those terms in different ways now because homogeny didn't take on, and we use that term for homology. And so what Lancaster argued is that there's only been one history of a tree of life. And so all organisms, and therefore all features of organisms, share some degree of relationship and similarity one to another. And that was what he was trying to get at with these concepts. Very close relationship or more distant relationship. So evolution, as through Lancaster and subsequently, has made this enormous contribution to homology by separating out degree of relatedness and its importance in looking at homologous characters. Evo-devo, I'm going to argue, has made a contribution to this as well by dealing probably more extensively than any other aspect of biology with the concept of novelty. And the last part of the talk is about several examples of novelty. And so I'm using novelty in the sense of being not homologous. This is looking at this issue of if it's not homologous, what is it? It's analogous, it's homoplastic, or it's a novelty. And this is based on a definition that, before I get to the definition, I forgot I put in my postulate slide. What is novelty? I used to think I know, but I'm not so sure that I know anymore. But it's most certainly all about all the things that organismal and evolutionary biology cares about. Similarity, that is homology, relationships, phylogenetic history, and shared developmental and shared gene pathways or gene networks, which I've broadly called evolutionary history, which you could call it evo-devo or developmental genetics or whatever term you would use for this. Clearly these are major topics in organismic and evolutionary biology. And novelty is dependent on considerations of each of these components. So Guten, Wagner, and Gerd Mueller came up with a definition of novelty in 2003, a new constructional element in a body plan that neither has a homologous counterpart in the ancestral species nor in the same organism, to get around this notion of serial homology, like the vertebrae, which are serially homologous. So if there's a new constructional element, so they were thinking in terms of developmental processes, that you don't find in the ancestors, then that's novelty, because it's not homologous with something in the ancestor. Therefore it's novel, it's new. I modified that a little bit because I thought this placed a bit too much emphasis on the way the element was constructed. And so my definition was more or less the same, except that for me a novelty was a structure, a function, or a behavior is a new feature in a group of organisms that isn't homologous to a feature in an ancestral taxon. So working with this concept of novelty, something that's not homologous, we can ask, what do we really mean by this notion of novelty? The Western Hotel in Halifax, I don't know whether you may have stayed there, yeah, wonderful hotel in Halifax. Western Hotel in Halifax knows what novelty is. The hotel invites guests to pick the price of a room. This is the first time that we are trying something like this, and as far as we know, it's the first time a hotel has attempted this, so it is certainly novel. So that gets the notion across quite nicely, I think. It's not homologous to anything we've done before. It's something new. It's something that you wouldn't have expected from past history. It's novel. Now that's tricky in evolution, because evolution works with things that are already present. They may be genes, they may be structures, they may be chromosomes, they may be whatever. So how can we talk about something that doesn't have a homologue in the ancestors? Where's it going to come from, and what's it going to be like? Okay, this is not an example of novelty. This individual has all of the features that were present in a close ancestral relative, the chimpanzee. So this is not an example of novelty, because a novelty is a new feature in a group of organisms. It isn't homologous to a feature in an ancestral taxon. These features are all modified from the ancestor, but not novel. Okay, so I want to look through 3 examples of novelty at 3 different levels, just to take us to the end. One is the origin of the neural crest. A lot of people, particularly Marion Bronner-Fraser, Marion Bronner as she is now, and a number of other labs, have asked about the evolutionary origin of the neural crest. Now, I've mentioned the neural crest in passing. That's where the craniofacial skeleton comes from. It comes from the neural crest. It's the embryonic origin of the exoskeleton, of the teeth, and the clavicle. And so it's a novelty of vertebrates, because it's not present in non-vertebrate chordates, it's not present in neurochordates, it's not present in ascidians. And so the neural crest then, I'm not sure which is the best way to, maybe this one. The neural crest is this region of the neural tube is developing from this flat plate of neural ectoderm. And as the neural tube folds up, you have these population of cells in green, and that's the neural fold, because it's the fold of the neural crest. And these cells, as the neural tube closes over, these cells migrate, and they migrate from the brain, they migrate from the spinal cord, so they migrate all over the body. So it's this population of cells that has an origin in the neural ectoderm. The ectoderm is going to make the neural tube, and that migrate all over the body and produce an enormous diversity of structures, more than any other part of the embryo, more than the mesoderm. There are more cell types that come out of the neural crest than come from mesoderm. From the trunk, you get pigment cells, you get sensory neurons and ganglia, you get sympathetic and adrenal gland cells. From the cranial crest, you get cartilage, you get bone, you get connective tissue, you get teeth, you get some muscle elements. So an enormous diversity of tissues. So clearly when this evolved, when the neural crest evolved, a lot of potential was evolved for the development of a lot of tissues. Not necessarily in one jump, but the potential with these neural crest cells was there for an enormous diversification of cell types, because bone is a cell type that you don't find in any of the invertebrates. You find cartilage, but it's a very different sort of cartilage. You don't find dentin and enamel in any invertebrate or in any non-vertebrate chordate. So a whole variety of features that are typical for vertebrates evolved with the evolution of the neural crest. Okay, so how do you investigate the origin of what's been called a germ layer? The neural crest is the 4th germ layer. You look for homologs of the neural crest in the neural tube of ancestors, which, what I said that twice. Oh, sorry, you look for homologs of the neural crest, yeah, in the neural tube of ancestors. So you look for something like a neural fold. Or you look for homologs of neural crest cells in ancestors. But if this is a novelty, you're not supposed to be able to find anything in the ancestor that looks as if it would be part of the structure. Or you could look for homologs of neural crest derivatives, like cartilage in the ancestor. Or, because we can't get at the ancestor developmentally, so what we look at is living surrogates of what we think the ancestor might have been like. But of course the problem, as we've discussed, is that novelties are not supposed to have ancestral homologs, but everything has to evolve from something. So what do we go looking for when we try to look for the evolutionary basis of the neural crest as a germ plate? Well, the approach that's been taken takes into account two phylogenetic scenarios for where vertebrates came from. The vertebrates either arose from an Amphioxus-like animal, or vertebrates arose from an Ascidian-like animal. Those are the two major competing hypotheses for the nature of what that ancestral vertebrate, that ancestral chordate that became a vertebrate might have been like. It was either Amphioxus-like, or it was very different, Ascidian, with a larva that had a notochord, which makes it a chordate. Okay, so this is a slightly complicated slide, but I'll also go through it. So this is just to set out the scenario. If the ancestor was a cephalochordate, assuming it was like modern Amphioxus, or if the ancestor was a urochordate, if it was like a modern Ascidian. And so here we have vertebrates, and we're arguing that their closest ancestor are cephalochordates, and so the evolutionary changes that relate to the neural crest evolved in this lineage. Or we're looking at the evolutionary ancestor was an Ascidian and to a vertebrate, and therefore the ancestral changes occurred in this lineage. The two basic, for our purposes, the two basic different things about Amphioxus and Ascidians are that cephalochordates have mesodermal cartilage in their gill slits, but no neural crest cells. So these guys have cartilage, which is a neural crest derivative, but they don't have neural crest cells. Whereas Ascidians lack cartilage, but they have cells that have been identified as neural crest-like. They have cells which migrate out from the neural tube and which make pigment and connective tissue and neurons. So they behave the way you would expect primitive neural crest cells to behave. Whereas if the scenario is that this is more like the vertebrate ancestor, then we have to look at the origin of neural crest cells at some stage after the cephalochordates gave rise to the vertebrates. Okay, so we turn to look, the way this has been looked at is to look in, I'm just going to look at the Amphioxus studies, is to look for precursors of genes involved with the formation of a particular neural crest tissue, in this case cartilage. And so we use a derived cephalochordate Amphioxus. And it's obviously important to stress, although it's clear that this is not an ancestral vertebrate. This has had 450 million years of evolution or more. We use it as a surrogate for the vertebrate ancestor, which is really a bit of a stretch. Using any living organism as a surrogate for an ancestor is a stretch. And so it has a mesodermal brachial basket cartilage. And as we've seen, that gill arch cartilage in vertebrates is from neural crest. So there was a cartilage, there is a cartilage, but it's not from neural crest, it's from mesoderm. So that implies that this might, if this is anything like the ancestral condition, this might be an ancestral condition. Okay, so we need to look then for, well, I'll leave it. Okay, so this is one way in which this has been studied. And before I go through this, I think I'll just show you the conclusion on the next slide. These are genes involved in neural crest-derived cartilage. And the expression of this network of genes in Amphioxus. So these are genes in the cranial-neural crest of vertebrates that are involved in the formation of cartilage. And these are genes in different layers, the neural tube, the pharyngeal mesoderm, the notochord, the gut, in Amphioxus, that are in vertebrates involved in cartilage formation. And so these are studies from Marion Broder-Fraser. Let me just tell you her conclusion before we go back to this. No single Amphioxus tissue co-expresses all or most of these genes. However, most are variously co-expressed in mesodermal derivatives. So our results suggest that neural crest-derived cartilage evolved by serial co-option of genes which function primitively in the mesoderm. So it's this concept of co-option of genes within the organism from germ layers into the neural tube, which then became associated with neural crest. And so if we now just go back to this and we ask what are some of these genes, SOX9 is a very important gene in cartilage development, SOX genes. We find SOX genes expressed in mesoderm in Amphioxus. Collagen A, which is a homologue of the collagen which is found in vertebrate cartilage, is expressed in the notochord. Other genes, RUNX, which is very important in cartilage development in vertebrates, is expressed in the gut. And so you see where the concept comes from, that there were genes which were serving functions. Here's collagen A in the pharyngeal mesoderm, as well as in the notochord. That there are genes which were serving functions, which if they came together with other genes in a different configuration, could lead to the potential evolution of a new set of cells, or a new gene array, or a new set of cell types, in this case the neural crest. So it's this concept of co-option of genes from within the organism to make this new germ array. So gene co-option within the organism may underlie the evolution of the neural crest. I say may, this is a scenario. May underlie the evolution of the neural crest and neuronous cartilages as vertebrate novelties. Novelties in the sense that there weren't neural crest cells there, because the genes were present. Clearly you have to have some genetic basis for a structure to appear. So the morphological feature is novel, the neural crest, the neural folds, the neural crest cells, but the cellular and genetic basis existed. So do we still regard that as a novelty? Because clearly the machinery on which to base that novelty was we assume was present in the ancestor. Okay, the last big example I want to look at, and then one small example, is another way in which novelty can arise. And I realize that there's an expert in your department who works on plant cell walls. Is she here? Good. I mean, I can get away with this. Digestive plant products are the cotton root nematode. This is a nematode which invades the roots of cotton plants. And this wonderful study came out a couple of years ago. Multiple lateral gene transfers and duplications have promoted plant parasitism in nematodes. So the two concepts then are multiple lateral gene transfer and gene duplication. Okay, so here we have 60 genes and 6 protein families that degrade plant cell walls are in the genome of this nematode. 60 genes, 6 protein families. Pretty diverse range of enzymes to digest plant cell walls. Normally you need termites in the gut, or you need some other way of doing this. This is novel, although we don't have any detailed knowledge of the relationship to the ancestral taxa from which these genes arose. And there's a whole variety of different sorts of enzymes that I've just listed out here. And this slide I've just shown from this paper. The closest relative to these nematodes that has these particular classes of genes. And so in the case of some of these arabinases, it's actinomycetes. In the case of the cellulonases, it's some beetles. In the case of this xylanase, it's Clostridium. So a whole variety of diverse eukaryotes are the closest relative to these nematodes that have these particular genes involved in digesting plant cell walls. Okay, so where is the novelty? The novelty then is these multiple, presumably independent, because they came from so many different sources, assuming that the closest ancestors, relatives are the ones that provided the genes, multiple independent lateral gene transfers from different organisms, followed by many gene duplications to form these multiple gene families. So the function was already there, but gene duplication didn't have to be associated with new function. The functions were already there. The gene duplication gave this enormous potential to degrade the plant cell walls. So this is an example of the introduction of novel genetic information. And I have a couple of slides that just summarize that. So for the organization of the neural crest and for the origin of neural crest cartilage, we looked at gene co-option within the organism, followed presumably by some neo-functionalization, because collagen A1 was there, but wasn't involved in some of the features that collagen does in vertebrates. For the digestion of plant cell walls by nematode, that's lateral gene transfer, gene duplication, the gene functions are already established before the transfer. Okay, the last couple of minutes. So gene co-option and neo-functionalization, or lateral gene transfer and gene duplication. Another way in which I think you can get novelty is through new symbiotic relationships as a way of introducing the genetic diversity. You can't do it in your own genome, so you set up a symbiotic relationship with another organism, that enables you to do it. And let me just give you one example to end with, which I just discovered 2 or 3 years ago and is really neat. You probably all know it. Do you all know about Ossidex, a genus that eats whale bones in the bottom of the ocean? Does anybody know about that system? Good, good. Okay, so here are these species of Ossidex. It's a marine beardworm, this particular family. There were thought to be 5 or 6 species. There are now known to be 24 species. They colonize whale bone on the bottom of the ocean at depths of 3,000 meters or so. See, here are all these worms colonizing this whale bone. They're sexually dimorphic. The females are large, these worms, 50 millimeters long, here's a female, surrounded by a gelatinous capsule which is colonized by hundreds of dwarf males, which are non-feeding. So you have this large female colonized by these large numbers of males. You become a female if the embryo falls to the ocean bottom on a piece of whale bone, it becomes female. If it doesn't, it becomes male. It's one of those really weird sort of environmental sex determination things. The females lack a digestive system. So here they are living on and feeding off whale bone, but they don't have a digestive system. And the males certainly don't have a digestive system. They have virtually nothing. I mean, it actually looks like a nice life to me for a male. Okay, so their paper that just came out in 2013, How to Get into Bones, Proton Pumps and Carbonic Anhydrase in these Ossidex tube worms. Epithelial extensions, which are called roots by analogy to plants, from the posterior body of the female, penetrate the bone that the females are living on. And they branch and they form this extensive network of epithelial tubes filled with endosymbiotic bacteria. So we've got this symbiotic relationship here with bacteria. And this enormous surface area of the whale bone, which is exposed to the female by these epithelial extensions. These root epithelial cells produce very high amounts of VATPase and carbonic anhydrase, which create this incredibly acid environment in which the products of the bone are digested, particularly the protein and the lipid products of the bone are digested, and they're then metabolized by the bacteria and used by the female. So you have this formation of a particular enzyme system to create this acid environment, which is appropriate for digesting bone, and then you have the endosymbiotic relationship with the bacteria. The other place where you see these enzymes, this is in the epithelial extensions of the back end of a female worm. The other place where you see these enzymes is in the mononuclear osteoclasts of teleosts and the multinuclear osteoclasts of tetrapods, which produce the same sorts of enzymes and proton pumps in this highly specialized cell with these highly integrated and diffuse connections to the bone to dissolve bone in vertebrates. And so you have here a wonderful example of sort of parallel evolution of this enzyme system, which in the case of vertebrates takes the production of a very specialized population of cells from a particular cell lineage, and which in worms is done by the gut epithelium, by the epithelium from the back end of the body. So, organismal evolutionary biology is alive and well 3,000 meters beneath the oceans, as is in the soil with the nematodes, and as it is 90 meters above sea level in the University of Massachusetts in the graduate program in organismic and evolutionary biology, which has produced 60 PhD students and 30 MSc graduates over the last 20 years, 17 Darwin Fellows, 4 or 5 of whom talked to us today. And so, congratulations and happy birthday to OEB.